So we're going to review one more time all of your different organic macromolecules. So if you're under the organic notes, we are now looking at the handwritten notes. So we said the first time those typewritten notes going through, let those terms settle in. Let's come back, revisit these terms a little bit more, and hopefully now it helps to clarify a lot of stuff that you guys have. Also, a lot of your pictures for your flashcards. If you can't find them or you don't quite sure the pictures that I'm kind of looking for, or if you think the structures you have are too intense or too much, because a lot of you guys will type them in online and it will give you the hardest ones that you could possibly think of. You'll see some of them throughout here as well. So once again, write down questions, ask them in class. Please, please, please ask them in class so that we can go through them. Okay, so we know that there are 4 types of organic macromolecules. Our first one are our carbohydrates that we went through. Functions, they're basically your main source of energy. Okay, energy storage, they can be structural. That's more your polysaccharides. So we know that we store glycogen within our liver, and plants use cellulose, which is a structural polysaccharide within their cell walls. But their main source, first place your body wants to go to for its energy. We know that there are monosaccharides, disaccharides, and polysaccharides, single sugars, double sugars, and very large complex carbohydrates, or many of those little sugars joined together. Your monosaccharides, glucose, galactose, and fructose, we said they are isomers of each other. C6H12O6. All 3 of them have that formula, but when you draw them out, they're all a little bit different for you. So down below is glucose. Usually it's going to come in rings, your sugars will. Not all the time, sometimes you might see it linear, but 99% of the time it's probably going to be in a ring structure, which is how you can identify carbohydrates. Another way they will usually end in OSE, glucose, fructose, galactose, cellulose, amylose, and so on. The third way is their formula. They're going to be close to that 1 to 2 to 1 ratio that we usually will see. Remember, as you get larger and larger and larger in terms of those polysaccharides, the way that you're making them is that you have to take out water, H2Os. So you're losing H2Os there. So my formula is not exactly a 1 to 2 to 1 ratio the larger that molecule gets, but it's going to be pretty close to it. So there is glucose, there is fructose. Pretty easy to see a difference between them. They're both 6-carbon sugars. The only difference between glucose and galactose is this ending right here, hydroxyl and hydrogen. It actually is just flip-flop. So with your galactose, your OH or hydroxyl group is going to be on the top and your hydrogen is going to be on the bottom. Condensation reaction is just another term for dehydration synthesis. We usually hear it as dehydration synthesis. And we know that anytime you are making a larger molecule, you will actually take out water to create a bond. That bond's going to store potential energy in that larger molecule. That's basically what you see here. Monosaccharide plus a monosaccharide gives you a disaccharide. Notice the formula is not exactly double, meaning C6H12O6 plus C6H12O6 does not give you C12H24O12 because we had to take out the 2 hydrogens and an oxygen in order to create that bond. And that's why the ratio is not exactly double as you get larger and larger here. There's another example of it. Remember that dehydration synthesis means that you are putting things together. It's an anabolic reaction. Hydrolysis is the reverse reaction. You are breaking it down. Basic digestion is an example of hydrolysis or catabolic reaction. You're adding water, you're breaking the bond, you're releasing energy. And this is basically what's happening within your mitochondria through the process of cellular respiration. We know that since my monosaccharides are isomers of one another, my disaccharides are also isomers of each other. So if I take glucose plus glucose, it's maltose. Glucose plus galactose is lactose. Glucose and fructose is sucrose. So maltose, lactose, and sucrose are my disaccharides. They still end in OSE. They all have the formula C12H22O11. But if you drew them all out structurally, they would be a little bit different from one another. So we can take that disaccharide, we can add some water to it, and create 2 smaller monosaccharides. Basic digestion. Take a cracker, it's more of a larger polysaccharide, put it in your mouth, let it start breaking down. That's what you're doing. You're changing it from a starch to a sugar. And you guys might remember that from first year bio. You guys probably did that little testing of foods. You test a cracker the first time, it has no sugar in it. Second time, you kind of chew it up, mash it up. Well, the reason it had sugar in it second time is because you've taken that carbohydrate, broke it down to a disaccharide, broke it down into a monosaccharide. So now it does have sugar. You broke that large molecule starch down to a sugar. And the way that you guys did that or tested for that was Benedict solution. This is the same Benedict solution we use to test glucose or sugar for in your urine lab. So your monosaccharide with Benedict solution is going to kind of turn this kind of a browny red color, depending upon the Benedict solution that you would have. Usually we don't see yellow too much, but it's more like a brown or a brick red or an orange color that you would be there for that positive plus 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 sugar. You can do the same thing with your disaccharides. For polysaccharides, they're going to test negative for sugars because those sugars are giant molecules within your polysaccharides. So it's actually starch that you would test for there. So starch is polysaccharides. When you break that starch down, that's when you're getting your sugars. So starch, you would test for with iodine, and that you did do in first year bio. So you took that iodine, it's kind of like an orangey brown color, and when it comes in contact with starch, it turns black, kind of this brownish black color, it's like a purplish black color. Remember, we store our polysaccharides as glycogen within our liver. So all those excess sugar molecules in your body's going to start stringing all those glucoses together to put it away as that form of glycogen, large molecule, in the liver for when you do need it at other times. Other types that we have is amylose. We have, which is also an energy storage, but that's more for plants. We have structural polysaccharides such as cellulose, as well as chitin within fungus cell walls. Remember that this cellulose plant cell walls are not easily digested. So when you guys are eating these plant cell walls, we can't really break them down. We need help with them. So maybe some bacteria within our gut might do that. And we'll see some other organisms here on the next page. Starch is an important source of carbohydrates in the human diet. We eat carbohydrates when we need energy. So marathon runners, for instance, I said, the sports teams that do pasta dinners before a big game or a match, they're hoping to get energy for the next day. So if you're that marathon runner and you're eating lots of pasta, you're storing that away as glycogen within your liver so that you can kind of break those bonds as you're going through those 26 miles and doing that run. Some plants such as seeds, peas, beans, corn, these have really, really thick cellulose walls on them. The corn, we can use this cornstarch. It's actually a thickening agent, kind of like the pectin that we talked about, or the amylopectin that helps thicken jams and jellies. You can make things or things that have these starches within these large polysaccharides, pastas, rices, potatoes, bread, cereal. Those are things that we kind of want to eat in order to make sure that our bodies have the energy to get us through the day. Some other starches that we have, amylose and amylopectin. So amylose is more of a structural polysaccharide. Amylopectin is more like a thickening agent that we find within plants. Here's my amylose. It's just a whole bunch of polymers of glucose. Glucose plus glucose plus glucose. We bonded them all together by taking out some water, dehydration synthesis. And then you can reverse that and you could go through and break it down. So we could take this particular starch and begin to break it down. Notice my formula here. I don't really know what my formula is. Depends on how many are joined together, which is why I have that variable there. So we go through, we start to break that amylose. We use the enzyme amylase to help to break that molecule down. It gets a little smaller of a polysaccharide. We use another enzyme to break that down. We break it down to a disaccharide. Maltose, we use maltase, another enzyme. Remember we said enzymes are very specific. And it's going to keep going on until eventually we get down to our monosaccharide. Remember, every time you break one of these off, going through the process of hydrolysis, you are releasing energy. Lugol's iodine is what we said we use for starch. We just talked about the color change up above. Within us, we said animal starches, you guys have glycogen within our livers. It is a polymer of glucose, meaning it is a large molecule of little itty bitty glucoses put together. So you guys eat foods that contain fructose or sucrose within them, so your smaller carbohydrates, and you have glucose within your blood. So if you're going to use that right away, your body's going to take it into your mitochondria, the powerhouse of the cell, and it's going to burn it in the process of cellular respiration. And through that process, you release some carbon dioxide, you exhale it out, water vapor, which you also lose, and then ATP, which is your energy. That's why you need to eat glucose. If you don't utilize it right away and you have all that glucose within your blood, you have an enzyme within there, actually you have a protein within there that's going to help to do this, and it's going to be insulin. So insulin helps to regulate blood sugar. So it takes all that excess glucose that you have within your blood, it starts to string them together, those little itty bitty molecules, and it forms glycogen with it. It takes it, puts it in the liver until it needs to be released later. So that's what insulin is helping to do. Those people who cannot produce insulin, well, what happens now? I have excess sugar within my blood, and my blood sugar levels get really, really high, or at times they get extremely low, depending upon the medications and the way that this is produced. But a lot of times you're going to have high sugar levels because the insulin is not able to pull that glucose out of your blood and store it as glycogen within your liver. Here's some more for you guys. Like I said, I'm just going to kind of run through, but this shows your cellulose. So this could be an example. I have open bonds at the end. So there's usually a heck of a lot more than what we have here. So I have a whole bunch of these particular chains put together of my glucose, alternating alpha and beta. I'm not going to say, hey, what is this particular structure? Do I have alpha or beta? I just want you guys to realize alpha and beta. Are my bonds facing down? Are my bonds facing up? And that makes a difference on what molecule we're looking at. That's all. So where it looks like a bunch of rings put together, up bonds, down bonds, things like that, are going to change the carbohydrate that we're looking at. These linear chains, these huge molecules, are very, very strong. We said that some organisms have trouble digesting it on their own, and they need to rely upon bacteria, such as the cows, as well as termites, who eat a lot of this cellulose material between termites feeding on wood, cows feeding on grass and hay. They need bacteria to help break it down so that they can get the nutrients from it. Cellulose, for instance, is about 2,000 to 3,000 glucose molecules strung together. So we're talking really rigid, really strong, really hard to break down. So if I said that and you guys are giving me glucose, or cellulose, which I don't need 2,000 to 3,000 units, and I definitely don't need you counting up 2,000 to 3,000 carbon, hydrogens, and oxygens within those structures, that would be the formula that you would use for me. So depending upon how many we have, there's my variable. Fats, oils, and waxes, we talked about triglycerides. So here is my glycerol molecule, my backbone, C3H8O3, fatty acid chain number 1, fatty acid chain number 2, fatty acid chain number 3. So you're going to go, well, that looks a little different than the one we looked at before. Those R's that you represent are looking at 3 different hydrocarbon chains. So it's a hydrocarbon chain with a carboxyl group, hydrocarbon chain with a carboxyl group, hydrocarbon chain with a carboxyl group. I'm saying that each one of them can be a little bit different. So maybe this R is 400 carbons with hydrogens. Maybe this one's 50 carbons with a double bond in there and some hydrogens. Maybe this is 5 carbons long. So each one's a little bit different. But I know that in order to join them together to this glycerol, I need to remove 1 water for each of my chains that I'm adding on. So since it's a triglyceride, I have 3 chains. I'm taking out 3 waters. I'm making this super big triglyceride molecule and showing the 3 waters that I'm taking out through the process of dehydration synthesis. Same process as carbohydrates. Same process if I'm digesting it down and I want to go through and eat this. I'm going to add 3 waters, break the bonds, get the energy from it. Here's some examples of your fatty acid chains. So oleic acid is actually a saturated acid. Remember, a saturated acid has at least 1 double or triple bond within it, and there's my double bond there, which means because I am sharing 2 pairs of electrons between these 2 carbons, I have no place for hydrogens to attach here. This unsaturated fatty acid chain is healthy for you. Usually it comes from plant sources, olive oils, canola oils. They're usually liquid at room temperature. If I look at palmitic fatty acid chain, this is my saturated one. So remember, saturated, think of like when the ground is saturated, you can't hold any more water. This chain is saturated. I can't hold any more hydrogens. And that means that I have all single bonds between my carbons. So here is my hydrocarbon chain. Here is my carboxyl group. This is the ending that's circled where I'm going to bond it to that glycerol molecule by taking out water. Your saturated fats usually are going to be solid at room temperature. They're usually going to come from animal sources. Some characteristics of fats: they are insoluble in water, which means they don't dissolve. You can't put them in there and they're just going to kind of break apart. If you take sugar and kind of mix it with water, yeah, they're going to start to break apart. However, these guys are hydrophobic. They have a fear of water and they will not dissolve. Waxes, we said, are protective waxes. They're easily molded, so they can kind of shape them because they're solids. We find them on plant leaves and stems. It's going to prevent dehydration. It's also going to prevent or protect them from insect invasion. Some animal waxes, they're more or less protective coatings, such as beeswax. It actually comes from wax glands that are located on the bees. We have sperm whale blubber. We have lanolin that's on wool. We have earwax within our ears that is also protection. So now let's take that molecule, that triglyceride, and take off one of our fatty acid chains. So here we go. We have our glycerol molecule, fatty acid chain number 1, fatty acid chain number 2. I'm pulling off that 3rd chain. So instead, now I'm going to put on a sugar. Maybe it's glucose, maybe it's galactose, and here's one of them for you. So here's glycerol, fatty acid chain 1, fatty acid chain number 2, and now I put on this particular sugar molecule. This is a glycolipid. This particular glycolipid or sugar lipid that we see here is going to help with transmission of nerve impulses. Some glycolipids are also found within the cell membrane. Okay. Next, let's change it up a little bit more. Glycerol, fatty acid chain number 1, fatty acid chain number 2. Now instead of that sugar or through that third fatty acid chain, now let's put a phosphate group. There's my phosphate group right there. That phosphate group is my head portion, my PO4 group. My fatty acid tails that you see here are these two little squiggly tails at the end. They are connected by that glycerol molecule, dehydration synthesis. This is your phospholipid. So when two of them come together tail to tail, you are creating that phospholipid bilayer that makes up the cell membrane. Remember we said this phosphate head is polar. It loves to be near water. It is hydrophilic. Your fatty acid tails, anything that's fat is going to be hydrophobic, doesn't want to mix with it, has a fear of water, and it has a non-polar end. So when we create a bilayer, my tails are going to be touching together in the middle, and they're kind of hiding away from water, where the head portions are facing the outside of the cell and the inside of the cell, which there is a lot of water. So there's kind of an expanded view of it. Cytoplasm is in the middle, mostly water. So all my reactions are taking place. Outside, mostly water, depending upon where that cell is. Other types of lipids, we have steroids. Here's my little steroid nucleus, A, B, C, D, rings. You realize that those rings are made of carbons and hydrogens and oxygens. And whatever I put off of that, whatever functional group or ending that I put off of that steroid nucleus is going to change what I'm looking at in terms of steroids. So once again, they're still lipids. They're still going to be insoluble. They're still going to be hydrophobic. And they're still going to be non-polar, just like those fatty acid tails that we had. So here's my little steroid nucleus. To that, I'm adding this particular chain of carbon and hydrogens. I'm adding a few more carbon, hydrogens, and a hydroxyl group off of here. This particular structure that you see here, if you need it for the back of your card still, is cholesterol. And we say cholesterol is synthesized or created in the liver. And it's also found within cell membranes. It helps to make up the myelin sheaths of certain nerves within your body. And there's two forms of it, HDL and LDL. HDL, high density lipoprotein, is what we're going to find within our cell membrane. We like that type of cholesterol. That's going to help to be kind of like holding onto receptors, helping move things through. And that's why we say our cell membrane is a phospholipid bilayer that is fluid mosaic molecules made up of a bunch of different pieces, not just phospholipids. Your low density lipoprotein, LDLs, is your bad cholesterol. You want to keep that level low because that can tend to lead to building up within the arteries, leads to arteriosclerosis, which in turn can lead to heart attack and stroke. I can take that steroid nucleus. Here's testosterone, here's estrogen. All I'm doing, I have the same nucleus. I'm changing my elements that go around it. So there are the sex hormones for both male and female, still utilizing those steroid nucleuses. Proteins, type number 3. So we start off with functions. We went through a lot of these. We said structural. We talked about collagen within the skin. Also, another type of structure, we find proteins within viruses. They actually have a protein coat that surrounds them. So we don't really think about viruses that much because it is a non-living thing. And we'll see in the next one as we work our way into cells, even though it's not a real living cell. But as we work our way into bacteria, we'll touch real quick on viruses and how they're a little different from one another because they do have proteins within them. They do have nucleic acids within them, but they're not a living thing. They're not a cell. We said they can be regulatory, such as hormones, such as insulin. We just saw it above when we were looking at our blood glucoses. And if you have glucose within your blood, it's the insulin that's going to kind of pull all that glucose together and store it away as glycogen within the liver. They are contractile, actin and myosin, the fibers or the bands within your muscles, transport hemoglobin, myoglobin. You guys did the protein lab on it. Storage, protective. They help to make your antibodies. Really important this time of year, especially this year more than ever. They help to fight infections within your body. They help to make up your cell membranes. You have protein channels. You have little receptors. They make up toxins such as bee stings, jellyfish stings, snake venom, botulism from bacteria, and they help to make your enzymes. They are your enzymes. Enzymes are special types of proteins. They speed up reactions. They usually end in ASE. They can be used over and over and over again, and they're specific. So you see sucrase there. It only works to break down sucrose or build up sucrose. Lactase is for lactose. Maltase is for maltose. usually ends in ASE. Pepsin, there's your exception. It actually helps to break down some proteins within your stomach. Here's my structure. We've already looked at essential carbon. Along each side of your essential carbon, we said first is your amino group. That amino group is on the opposite side from your carboxyl group. That's COOH group. Two functional groups that we looked at the very beginning of these notes. Around that central carbon, you'll have a hydrogen. Around that central carbon, you'll have an R group. This is not an element. It's just saying this is the only part of this entire structure that's going to change. It's one of 20 different endings, giving you one of 20 different amino acids that when put together, they can then create unlimited number of proteins. So that R group, we said polar and is uncharged is 7, pH of 7. It can be nonpolar. If it's acidic, it's negatively charged at pH of 7. If it's basic, we have a positive charge at a pH of 7. So usually what you're going to see is that your COOH group, your carboxyl group, has more of that negative charge, has more of an acidic end to it, and your amino has more of a basic end to it. Amino acids are known by their common names. I am not going to have you memorize 20 different amino acids when you can pull them up somewhere ever anymore. But just to give you an idea, because you've probably seen some of these before, methionine, alanine, leucine, glycine, serine, so on. Methionine is one that we'll really look at because that's the amino acid that basically starts all your proteins, the majority of your proteins. We create them the same way. Dehydration, synthesis. There's my amino group, there's my carboxyl group, there's my R group. I'm joining this amino acid together with this amino acid. I'm pulling out my water. There it is. I have a new bond. So this amino acid now joined together with this amino acid are joined by what is known as a peptide bond. Peptide bond is where we have removed water to join them together. This structure here is now called a dipeptide, two amino acids together. If I keep adding, let's say I take out this hydroxyl group and a hydrogen off the next amino acid, and I keep going, I'm creating a polypeptide, many amino acids joined together. Another name for polypeptide is just a protein. They mean the same thing. A protein is a whole bunch of amino acids joined together. We talked about levels. We said they go from simple to very complex. These particular proteins, if you are just a chain of amino acids kind of hanging out, peptide bonds together, this chain of amino acids can dictate or can be dictated by a gene, which is a segment of your DNA or RNA. It basically tells you what amino acids to put together. This primary structure is just a sequence of amino acids. So this is like where I said you guys would all be holding hands and you'd be walking around the hallway and you'd be this chain of amino acids or protein walking around the hallway. Secondary structure, when I take that chain and I start folding it, so I showed you guys the little pipe cleaner, that was a chain of amino acids primary structure. I wrapped it around itself. It made it look like a foam cord by the time we were done. That's your alpha helix. I could take a piece of paper and I could fold it back and forth. And I would also have a beta pleated sheet, which is what we have here. My tertiary structure, there's a little bit more folding going on, but most importantly, I'm going to start connecting these folds with disulfide bridges or sulfur bonds between them. I'll take my little alpha helix. And what happens here is that sulfide bonds are going to start joining between these little curves that we have here. That's a tertiary structure. So we're going to get a little more complex as we move along. Your quaternary structure, globular, huge, giant mess. Usually it's 2 to 4 of either your betas or your alphas joined together. So for instance, hemoglobin is 2 of your alpha helixes and 2 beta pleated sheets, and they're all joined together in this huge giant globular mess. It is a giant protein. Your collagen can either be a tertiary or quaternary as well. You can see some of these other ones that are in there. So they can be fibrous, such as what we see within our muscles. Usually if they're fibrous, you're going to see repeating structures. So if you think like muscle bands and the strands within those muscles, that means we have fibrous proteins there. Other ones are globular proteins, hemoglobin. If you guys remember doing that protein lab, it barely is moving out of those wells. It is super duper heavy. It's a big, giant globular protein. Remember what it does? It carries oxygen within the blood. It's going to be a hemi group or an iron group, which means basically a binding protein. It is binding oxygen and it is transporting it throughout the body. So when you are anemic, what happens is you have low hemoglobin, you don't have as much oxygen, or you don't have as much of this protein to bind oxygen and transport to your body. So you tend to be a little bit more tired because you need oxygen to get those muscles to make your energy for you. That's the whole purpose of cell respiration and producing that ATP, aerobic respiration. It makes more energy than your anaerobic respiration. 36 to 38 ATP versus your 2 molecules of ATP. So it makes a big difference getting that oxygen to those particular areas. Analysis of these particular ones, tools to use for analysis as well as identification of different macromolecules, chromatography. You can do chromatography with amino acids. We're still looking at solubility and mass, which is what we did with our chromatography lab. We didn't do amino acids. You guys did black marker, but we still spread things out or separated things out based on solubility and mass. Electrophoresis is what you guys did. So we still have mass as well as molecular mass as well as charge. Heavier molecules stay closer to the origin. Lighter molecules travel farther, and then you have positive and negative charges. Yes, you need to know some of the gel electrophoresis stuff for your test. So my cathode was the black end of the box, my anode was the red end of the box. Opposites are going to attract. So if my cathode is negative, my positive cations are going to go towards it. So opposites are going to attract. Know that anode and cathode. Know what charges go to it. Know how things separate within that gel electrophoresis chamber. Some of the other ones, nuclear MRIs, lasers, crystallography, things like that. You might see some of those in college. It's another way just basically to analyze different types of organic macromolecules. Leads us to our last one. And like I said, we do this one pretty quick because you will have a whole section on it. But it's your nucleotides, which make up your nucleic acids. And once again, they are made up of one sugar, one phosphate, one nitrogen base. Or as it says here, one nitrogen base, one phosphate, one sugar. Do it backwards for you. Your nitrogen bases in DNA, adenine, guanine, thymine, cytosine, A, T, C, and G, and they go together in a certain fashion. So we know that adenine will always pair with thymine, guanine will always pair with cytosine. That's just something you're going to have to memorize. I always join one purine with one pyrimidine. So I showed you some pictures on your PowerPoint that's attached to this particular tab as well. Purine, as you guys see here, is a double ring structure. A pyrimidine is a single ring structure. So when I put them together, the rungs of my ladder should be 3 rings across. One purine, adenine, one pyrimidine, thymine, 3 rings across. I know which ones go together because adenine and thymine have 2 hydrogen bonds between them. Guanine and cytosine have 3 hydrogen bonds between them. That way, it's the only way that those particular base pairs, nitrogen base pairs, can stick together to make the rungs of your ladder. In RNA, everything's the same except thymine gets replaced by uracil. It does everything that uracil does. So it still bonds with adenine with 2 hydrogen bonds. It's still a pyrimidine, meaning it's a single ring structure, just like thymine does. It's just that we don't have thymine in RNA and we don't have uracil in DNA. Phosphate group is that PO4 group. So that's a functional group that we talked about. It's kind of a high energy group. It's going to create a high energy bond. It's going to bond the sugars and the phosphates to create the backbone of your DNA molecule. Last part of a nucleotide are my sugars. My sugar in DNA, deoxyribose, it's a 5-carbon sugar. It ends in OSE. That's how I know it's a sugar. We just said sugars end in OSE usually. Ribose is the sugar in RNA, so just slightly different sugars. They're actually isomers of one another as well. So pentose just means it's a 5-carbon sugar. There's one sugar, one phosphate, one nitrogen base are my basic structures for DNA, for RNA, and for ATP. Except ATP has 3 phosphates, adenosine triphosphate. DNA is deoxyribonucleic acid. It's huge. It is double helix. It's a double strand that's kind of twisted all together. So if I kind of took these 2 pipe cleaners and said, oh, look, here's my DNA, and then we twist them together, it is a double-stranded molecule as what we see there. So it's a pretty big structure. It makes up your chromosomes. It's found in the nucleus of every one of the cells that you have. And it actually carries your genetic code. So it has A's and T's and C's and G's. And the way that those A's and T's and C's and G's make everything about you, makes every trait that you have. It also codes for every protein within your body. So it's the code that tells your body how to put together amino acids to make the proteins that you need. Here's the problem though. Proteins are made way out in the cytoplasm. DNA can't get out of the nucleus. We need to rely upon RNA. So RNA is single-stranded. So it's going to take DNA's code and kind of copy it into a single strand. And because it's a single strand, it can get out of the nucleus, go out into the cytoplasm where it's able to hook up to a ribosome. Once at a ribosome kind of hooks onto it. and he starts reading the code on my messenger RNA that came from DNA. And as he's reading that code, he's kind of yelling out so transfer RNA can come in and bring the appropriate amino acid for them to start stringing together to make my protein for me. So there's 3 types of RNA that are involved. Messenger RNA, that's the one that takes DNA's message from the nucleus to the cytoplasm. He makes a single strand. Your rRNA is kind of the one everybody forgets about, and what he does is your ribosomes, everybody thinks of like these little balls in the cytoplasm. They're actually 2 subunits that need to be kind of glued together and activated so that my messenger RNA strand kind of gets pulled through that ribosome. Transfer RNA, I think of him as like a little taxi cab. He carries your amino acids to the ribosome, attaches them to the messenger RNA strand so that they can kind of match up together so that we're able to make the protein that we need. If we're talking about ATP, we have adenosine triphosphate. There is my nitrogen base. I've got some sugar in there, and I've got some phosphates. The more phosphates you have, the more energy you have. GTP is just an alternative form of energy. It's guanine triphosphate. So once again, it's just a different nitrogen base within that. So this kind of finishes up everything that you guys have. We'll do a little practice quiz. I'm not going to grade it, but I do want to go through it so that you guys have a chance to see how questions can be asked before we get to that organic test, 24th and 25th of November.